Another way of selecting genes is by gene set enrichment analysis. And in that case, so far, this is a lecture on gene set analysis, which comprises of basically those two methods, overrepresentation and the gene set enrichment analysis. And for the GSEA, we have is a 2-sample comparison. That's what you need to think about. So if you have 3 samples, this might not work. So comparing, for example, class A or B and B, so like let's say 2 cell types, or maybe in one cluster you have healthy control versus disease, and then you sort all those genes based on fold change. So the lowest fold change for class B to the highest fold change. And here in the gene set, let's say I list all the genes that belong to interferon signaling. So in here, you have a black bar that is highlighting one of the genes from the pathway. So like here, you have all the genes that are highlighted in this gene set. So what it does is try to find by random walk the maximum distance from those genes. So you can see that some of those genes are always in the beginning here. So there are more genes in the beginning compared to the end. So this gives you a more enriched score towards this end. So that means that these genes are more expressed in class A compared to class B. And then you actually do a test of that compared to a random distribution, where every single gene will be randomly distributed or uniformly distributed across all this sorted gene list. And because you would do this rank based on fold change, then actually you don't need to add any filter to your method. You just can do differential gene expression or calculate the fold changes or a p-value, and just use that directly into the test, because those fold changes will also be considered. So if you take this example, if you look at this curve, you see that this one is enriched to one of the samples here, because you see many genes are more in this side than on the other side. This one is also enriched, is another condition, that more in this side than in this side. But this condition here is not enriched. You can see like this wave sign, but you can see that most of the genes are uniformly distributed all over. So this is basically the result of a gene set enrichment test. And of course, you have a p-value and enrichment score that you also can plot as a bar, for example.